Welcome to this edition of Research Connection. Joining me today is Dr. Megan Schreeg. Thanks for joining me.
Yeah, no problem.
So tell us a little bit about yourself.
So I am a third-year anatomic pathology resident here at NC State, I have been here actually at this school for 10 years, I think now. I first came here as a part of the DVM PhD program, and then stayed for my residency as well. I really enjoy research of feline diseases. So I did my PhD studying cytozoan felis with Adam Birkenhauer, who's one of the internists here. And then when I was interested in doing my residency here, I talked to Dr. Jody Gukin, who is another internist here, because I also knew that she studied cat diseases, and said, hey, what kind of cat diseases can I study during my residency? And so that kind of started this research project that I am going to talk to you about today.
And this research project led to an award.
Yes, it did, surprisingly enough. So, yeah.
And what was the award?
So at the annual ACVP Pathology Conference, which was held this past November in San Antonio, they have Young Investigator Awards, and they give first through third place for a couple different categories. There's like a natural disease category, experimental disease category, and so in the natural disease category, I got second place for this for my poster presentation of this project. And what was also really cool is the first place winner was actually another NC State alum, Dr. Elizabeth Alloway, who is another just finished her anatomic path residency last year.
Congratulations.
Thank you, thank you.
So tell us a little bit about the research.
Yeah, so when I first started the residency, I just sat down with Dr. Gukin and I said, hey, I want to study some cat diseases, and she is well known and renowned for her expertise and knowledge on gastrointestinal diseases and hepatic diseases in both dogs and cats, and we both just talked about, hey, there's this syndrome that cats get, and it's called triaditis. And literally that's just, the tri is just referring to this inflammation that cats get of their pancreas, their biliary tree in their liver, and their intestines. And it's always been speculated that cats get this disease based on the anatomy that they have at that location. Their bile ducts and their pancreas ducts just kind of come in together and enter in the small intestine. And since all those organs are interconnected, it's thought that they're just predisposed to getting ascending bacterial infections into those organs and just concurrent inflammation. But what's really cool is, like, we were taught this is the anatomy that cats have at that location in anatomy in vet school. But then when you look into the literature, like, nobody's actually looked that closely at what does that location look like. So yeah, so Dr. Gukin's like, yeah, I'm interested in triaditis, and I'm like, what's happening at that location? And we're like, let's just look at it histologically and see what it looks like. And so then that spawned this project where what we ended up doing is basically any cat that came through the necropsy floor, we just said, well, let's collect tissues from that site and see what it looks like. So it just started out as this descriptive study, like, what does that location look like in cats? And so then that just became this big project where then we started also looking at, well, let's look at the liver in those cats and the pancreas in those cats and then all the other sites in the intestine as well, and see if we can find any associations between inflammation and those sites. So then today, like, now I've got tissues from 120 cats that we've looked at. I presented the summary of 80 cats at the conference, and what we found is that site does look like it's really important for this disease. So the first thing that we found is when we look at the histologic anatomy at that site, it's really complicated. It's not just like two ducts that join together. It's this really convoluted network of these ducts. And actually what's really cool is when you go back to the literature and look too, like, that's what it looks like in humans. So it's neat because maybe, studying this site could have implications for human diseases at that location as well. So that's the first thing. The second thing we found is there's a lot of just looks like lymphoid tissue in that area. So that also kind of seems to imply that activation of the immune response at that location and monitoring for potentially infectious agents also might be important.
Last question. Do you have any cats?
I do. I have 2 cats, and since you asked, I guess I'll tell you all about them. My older cat, Pip, is a tortoiseshell, and she is super sassy, as all tortoiseshells are. And then my second cat, Rue, she is a little black domestic short hair, and she is actually a survivor of cytozoan felis, this disease that I studied. She was a laboratory cat in a study that I participated in, and we infected her with cytosine, but then we cured her of it. So she kind of contributed a lot of knowledge to the knowledge base about that disease, and now she lives with me.
That's fantastic. Thank you for sharing.
Yeah, of course.
And best of luck.
Thank you.